episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show with another really fascinating guest today, helping to create a better tomorrow on many different fronts. Today, we have the honor of being joined by Dr. Karoline Kaid, who is co-founder and chief science officer at a company called Viro Biotherapeutics, which is a biotech company focused on developing cancer-killing or oncolytic viruses, ultimately designed to target pediatric and adult brain cancers. Dr. Kaid is also a postdoctoral fellow at the Center for Human Genome and Stem Cell Studies, which is located at the University of Sao Paulo in Brazil. It's a public university in the Sao Paulo state and one of the largest Brazilian public universities and the country's most prestigious. Dr. Kaid has her bachelor's in biology and genetics, her master's degree in the biology of cancer, and a Ph.D. in cancer genetics, all from the University of Sao Paulo, and has completed over a decade of experience in cancer biology research, including that focused on CNS tumor research, tumor stem cells, as well as other advanced therapies. As a Ph.D. student, she worked under the mentorship of Dr. Oswaldo Keith, where she acquired a broad background in preclinical models, and specifically in expertise in cell culture, brain histology, molecular genetics, stereology, among other disciplines. Her PhD thesis, which was awarded Brazil's Best in 2020, resulted in a number of patent applications and preclinical findings focused on future translational studies related to both biomarkers of aggressive stem-like tumor cells, ultimately revealing interesting therapeutic approaches using microRNA inhibitors as well as the oncolytic Zika virus, both focused on targeting stem-like tumors that could benefit patients affected by CNS tumors lacking current effective treatment. Recently, as a postdoc fellow at the Human Genome and Stem Cell Research Center under the guidance of Dr. Maiana Zatz, another luminary in the Brazilian biotech scene, she continues to investigate a variety of alternative immunotherapies with a focus on oncolytic viruses and is coordinating veterinary clinical trials using oncolytic Zika virus in dogs with advanced brain tumors currently. Dr. Karoline Kaid, thank you so much for taking the time to come on the show today.
Thank you. Thank you for the kind introduction. Coming from me for new seems better than I thought.
Well, yeah, it's you have a fascinating background in a variety of domains. Before we get into our core topics of the day, I'd love to hand you the microphone for a little bit, just to talk a little bit more about yourself. If you could sort of introduce us to you, everything from where you grew up, how you got interested in science and biology, and ultimately cancer genetics. I think that'd be a great way to start things off.
Okay. So since I was a kid, I was always fascinated about science and everything. Here in Brazil, we have a magazine for our kids, and my parents used to buy it, and I have two brothers. And when the magazine arrived, every month we were like fighting to be the first to get the magazine and read. And my brothers are all scientists. My biggest brother is from chemistry, and my little one is physicist. So we are all in this, and we were all curious about it. So since I was a kid, I wanted to be a scientist. And biology, for me, it's very fascinating because it's complex, and it's like amazing how all the mechanisms works, and we see this kind of how can I say, like a very fine adjust of everything. So I here in Brazil, I entered the university. We were not a rich family, So I had to study a lot here in Brazil at the best university, at the public one, but it's very difficult because we have a lot of people trying to get this university, and I didn't study in like a rich high school, so I had to study at home a lot, like 12 hours a day, and I got my my fellow to the university. And I think when I got here in the University of Sao Paulo, everything started. So since the beginning, I went to the lab and started my doing science, and then never stopped it.
Outstanding. Outstanding. It's a
Yeah.
it's a fascinating story, and watching. I had the opportunity to sort of go through PubMed and look at sort of the progression of your work over the years. And one of the I guess before we get into the core theme of oncolytic viruses and some of what you're doing there, a lot of your work prior to working with the oncolytic viruses was focusing on various aspects of stem cells.
Yeah.
And different properties, whether you've written about the proteome, the microRNAome, looking at pluripotency factors and things of this nature. And I think these are going to be interesting to talk about once we get into stem cell properties of cancer. But talk a little bit about what interested you in some of these areas, because they're really hot nowadays in terms of not just how we create stem cells or properties of stem cells, but the very unique connections between a stem cell and then cancer cells as well. So talk a little bit about your work, your earlier work in these stem-like property areas, if you would.
Yeah, so during the university, I always like it like stem cell, because we know that from one cell we can differentiate for all the other kinds of cells in cancer. And cancer is always like very fascinating, because we can, there's a unique mechanism and so dangerous and so unique in our body. And we have a lot of hallmarks of cancer that can do all this aggressiveness. And when the first time in college, I saw that it would be similarities between stem cell and cancer, for me was like, what? How? And then I started studying, and then I discovered that there's a lab here that's the main goal of Professor Oswaldo Okamoto laboratory. And there I figured out that there's a subpopulation of cells in cancer with stem cell factors. So there's some cells that has some factors. It's not a stem cell, but can proliferate a lot, can have the ability to renew. And these cells are the key for aggressiveness, for example, because they are resistant to the chemotherapy and all the conventional therapy. So imagine a tumor that after therapy, all cells dies, but that population is still alive, can proliferate, and is responsible for all the growth again of the tumor, and it becomes more aggressive. So there is these are the cancer stem cell theory, and there's a lot of good labs and scientists trying to investigate, understand this mechanism, how tumor has this kind of cell subpopulation, and trying to develop a therapy focus on these cells. And when we studied pediatric tumors, there a special thing on it, because these kids seems that these tumors origins from these cells. So it's stem cells that has the tumor transformation, neoplastic transformation, and it turns to tumor. So these tumor cells carries all these stem cells factors, and it makes these tumors very aggressive with a very poor prognosis. So that's why we always try to figure out and identify some biomarkers that are high expressed in stem cells and also in aggressive tumors. So for example, in medulloblastoma, it's a pediatric cancer with high incidence in kids with 4 years old, for example. There's a protein called OCT4 that is high expressed in stem cells, including, for example, it's used to reprogram these cells to a pluripotent stage. And we see the expression of this OCT4 high in kinds of patients that have very poor prognosis. So it can be used for biomarker to identify these patients that will have a poor prognosis, because sometimes the beginning they don't have these factors and the doctors don't see that they would be a metastasis or anything, but they have in this beginning the expression of these stem factors. And then if we identify in the beginning, we can properly treat this patient and not wait the metastasis in the aggressive stage, because after that it's difficult to treat these patients. So that's the beginning of all this. And also to identify biomarkers, we can use these stem mechanisms to stop it and think about treatment, targets and drugs to inhibit these, silence these stem mechanisms.
Excellent. And, now taking a small shift to the side for a moment, and we'll introduce the concept of oncolytic viruses. And this is not the first time on the show we've talked about sort of, I know we're in the middle of a pandemic, but sort of good things that viruses can do for us. We've talked a little about bacteriophages on previous episodes that like to, viruses that like to chew and lice nasty bacteria. Here we have viruses that preferentially like to infect and destroy cancer cells. And this is an interesting area that, it's been around for sort of in the literature, I think going back to the 1960s or so. Talk a little bit about oncolytic viruses in general, but also when it comes now to sort of merging these things together, what we're talking about, what is it that is it the stem cell factors? Is it other things that you're studying in the biology of the cancer cells that make these viruses happy, or that make them like these cancer cells? What makes this specific virus, and we'll get to Zika and other things in a little bit, but what makes oncolytic viruses say, hey, I want to go to that cell because it's interesting to me, but not that?
Yeah, that's a good question. So, as you said, it's a new thing. All the oncolytic virus started in the last 15 years. There's a lot of study. We had the first oncolytic therapy in US approved by the FDA, it's the TVEC, using the herpes virus. And now, last month, we had the second one in Japan, approved for GBM, glioblastoma, and it's growing, but it's very striking, right? Because what happened in a normal cell, when the virus enters in the cells start a mechanism to inhibit the virus replication. So sometimes is mediated by interferon. So it's antiviral response. It's it's the defense of the cell, right? But a cancer cell, we have a lot of mutations,
Mm-hmm.
and mutations in interferons, and the cancer cells has these when it turned to a cancer cells, he stopped all this defense because the cells needs to proliferate and to move independent for its own, to do everything it wants without interfering the defense. So all these defenses are broken. So when a virus enters in these cells, in the cancer cell, because there is no defense anymore, that's why these tumor cells, the virus can replicate freely in the cells. So it replicates, and after some times, after a little time, there's a lot of virus inside of the cells, and the cells can hold it anymore and lyses the membrane. That's the oncolysis. That's why oncolytic effect. So, the main message here is when a cells turns tumor cells, it happens because it is able to turn off all the defenses. And the virus use these defenses, that are no defense, to replicate. But in a normal cell, all these defenses are okay, and the virus are not able to replicate. That's why.
Got it. And now, continuing along that, let's then move into Zika for a moment, because here, obviously a couple of years ago, the world knows the name Zika like it knows COVID nowadays. Member of the flavivirus family, and related to dengue and West Nile, things like that. And I saw a presentation of yours that sort of highlight different things that say, wow, this is from a perspective of a virus that's existing in nature, it has these properties, and you listed, it gets across the blood-brain barrier, it deals with cancer stem cell like resistance, and then another fact you mentioned in the presentation goes into the immune privileged nature of the brain, deals with that. So obviously Zika is a horrible thing, but you found here's a very sort of pharmacotherapeutic application for it. Talk a little bit about sort of the early stage. I know you did some work in embryonic tissues, in both in vitro and in vivo. What are some of the things you found out?
Yeah, I like to tell this story because it's a very unique story that we can tell how science works, right? So I was working with this pediatric cancer and doing some trying to understand the factors of these tumors, these pediatric tumors, and I did a microRNA, and I figured out that these cells has a expression profile very similar to NPC cells. There's the neuroprogenitor cells.
Okay.
So I was doing this research, and I saw, wow, these cells has the same expression profile as neuroprogenitor cells that are present in the early stage when the babies are in the belly yet, when we are having all the neural development. So these NPC cells, the neuroprogenitor cells, are only present in our body when we are fetus, right? And in the same time, our center was investigating the Zika virus, all the mechanisms, right? Because it happened here in Brazil in 2015, and our funding institutions called us and said, look, we have a problem here and we need to figure out, nobody knows about this new virus, and seems that here in Brazil, this virus has some particular mechanisms, because just here in Brazil, we saw this association with the virus and microcephaly, and the same as COVID pandemic, right? So all the scientists were called, and this is new. We need to figure out what's happened to try to avoid it. And Mariana Zat's laboratory was studying the cells. She went to the north of Brazil and she collected a blood sample of a lot of women and babies. And she figured out that there's some twins, dizygotic twins. So that one twin had microcephaly and the other not. So we collected the blood of these two twins, reprogrammed the cells to generate iPS cells, pluripotent cells, and after NPC cells and neurons here in the lab, and trying to infecting these cells and trying to understand the mechanism of Zika virus in these cells. And she saw that the virus were only capable to infect the neuroprogenitor cells, only in that stage, right? Yep. That's why it causes microcephaly. And I was presenting my work. We have like more than 20 professors, 20 PIs doing in our center, collaborating in our center, and we have meetings. So sometimes some students don't like these meetings. Yeah. I say you need to come to these meetings because we figure out what all the researchers, all the other PIs are doing. And was there when she figured out that we were studying these cancers, and they had a neuroprogenitor profile. And she was, wow, the Zika virus infects just these cells. And we knew that there is this new oncolytic virus. So we thought maybe we can turn this bad guy a good guy, an ally. So it was a simple experiment, a simple assay here in the lab. I took the cells I was working and I got the virus and I put the cells in contact. And as you said, I was studying these tumors a very long time and trying to figure out and trying to get good targets to treat these cells, but it's difficult to see anything that really works, because when we put these therapies or these drugs in contact of the cells, we don't see anything in the lab. But when we do the tests, we see, okay, it inhibited the proliferation a little bit, it increased a little bit the cell death, but nothing that we can see by our own eyes. But when I put the Zika virus in contact of the cells 3 days after the infection, all the cells were dead. All the cells were in suspension, and I was, oh my God, what's happening here? I got like so surprised that I got out of the room, the culture room, went to the Mariana Zat's room, and there was Professor Keith there as well. And I said, all the cells died. And say, what? Yes, all the cells died. And they come to see, and it was like amazing, the results. My Keith that studied in Harvard and studying cancer for like 20 years, never saw that as well. So that was the moment that I stopped my PhD. I saw like a big discovery, and I put all my effort and everything to trying to figure out what was in front of me and started studying. And so we did all the other tests, and we did the preclinical study, and I saw that the Zika virus was capable to kill also the metastatic sites in the mice model. Was amazing, because I thought I never saw like a total remission, because these tumors are so aggressive. When I put these human tumors in the brain of the mice, in 2 weeks the mice dies. And when I put the tumor, and after 1 week I treated with the virus, the Zika virus, there was total remission. So I also thought that the equipment that we do this test was broke. And I said, no, it's broke because it's impossible. And I called the technician, and he started to do tests, and no, it's not broken. And then I increased my number because I didn't believe in that. So I increased the number of the mice, and yes, I saw more total remissions, and it was true. So it's amazing. And now we are trying, after we tested in dogs. So when we got the cover of Cancer Research in June, there was a lot of media. So the news were no, the journal here in Brazil, and a vet called us saying that there's some dogs with the same CNS tumors. So we went to the dogs, started treating these dogs here in Brazil, and also we had amazing results. And as you said, I think the unique effect that we see in the Zika virus, what makes it unique, is because it has the capability to cross the blood-brain barrier.
Yep.
Something that precludes drug delivery when we have a brain tumor. So after all the advances we have here in treating tumors, we have the immunotherapies, the PD-1, the CAR-Ts, everything works, but not in CNS tumors.
Yep.
Because they have this main aspect that contributes to the poor prognosis, right?
Yep.
So one, as you said, one is they are isolated organ. It's possible to these drugs cross this barrier. We have also the cancer stem cells that we talked before. So the CNS tumors has a lot of cancer stem cells, glioblastoma. We see cancer stem cells in these tumors that are responsible to the resistance of conventional therapy. And also because the immune system is not work completely in the brain, right? So there is immune isolation of the CNS, the central nervous system, from the body. So that's why we call these tumors, I like this word, cold tumors. Because when we have a cold tumor, we have a tumor without immune cells,
Yep.
without macrophages, lymphocytes, everything.
Yep.
But these new immunotherapies need these cells the presence of these cells in the tumors to make the immune system work against the tumor. But there's this immune isolation of the brain and the other cells. So the brain tumors are cold tumors without these cells. So the immunotherapies do not work. But it's amazing how Zika virus can cross these three main aspects and make it work for CNS tumors. Because first, it crossed the blood-brain barrier. We see it in the epidemic of Zika virus. It targets especially these progenitor cells. That's why it causes microcephaly, because the targets of Zika virus, they like just the cancer stem cells. They don't like the other differentiated cells. And when the virus got in the tumor, it wakes the immune system that something is working there, something is wrong there, right? So we see because there's a virus in the brain and infecting the cancer stem cells. and the immune system recognize these, and we see lymphocytes, macrophages recruitment, and then it starts the anti-tumoral effect of these immune cells. So we turn the Zika virus is capable to turn a cold tumor a hot tumor.
Mm-hmm.
And it can be treated with all the other immunotherapies and now make it work.
Got it.
So
yeah, it's a very elegant story, and it's sort of an example of I know once again viruses have a bad connotation, but the concept that here's something that sort of nature has perfected for this bad purpose, and you're repurposing it for something really good here. And it's I looking at obviously, aside from now a researcher, you're also an entrepreneur, and you and Dr. Zatz and Dr. Keith and so forth have put together this new company, Viro, and you have a whole plan in terms of a pipeline, not just medulloblastoma, but glioblastoma, other CNS solid tumors, and then you have plans, as you were saying, for sort of combinations. You can see how this would enhance, say, a CAR T therapy with the cold hot. Talk for a couple minutes on sort of your transition, or what into like entrepreneur now, what the Brazilian sort of Sao Paulo biotech ecosystem has been like in terms of raising money and putting companies together. Talk about a little bit of your journey on that side, if you would.
Yeah, that's very new for me, but to make it a real drug for the patients, we need to take off of the university and put in the markets, in the business way, right? And we are 5 co-founders. So we have Professor Maiana Zatz, Keith, Oswaldo Kemoto, Keith, we call Keith, Dr. Keith. We have Dr. Luis Caires. He was the first author of the first paper that was published in Cancer and Nature Communications about Zika virus, the study of the twins, with Ernesto Goulart, the other doctor. So these are our team, and we got Hugo Cabrera. That is he's in the biotech environment. And he is helping us to get this technology to the patients. So we started a startup, but it's very difficult here in Brazil because talking with some collaboration in US, and they also said that it's difficult to do this transition from the university to the biotech environment, also in US, because the university, we do something in our way here, but when you go to business, there is a lot of difference. You need to, you talk to people differently. It's totally different. We have like a own code between scientists that we always collaborate. We always do things together, trying to figure out the problem. But when we go to business, we need to be careful about PI and IPs, right? So we need to do the patents, ideas, sell, NDAs, all the things that is new for me. But I see all the things as a tool, just a tool, the best tool to make all these possible. So I need these tools. There's no other way, no better other way. We need a startup, a company. We need all the big pharmas to help us to make it possible to help all the people that need this therapy. So to go global, right? To because there's a lot of people suffering with these tumors, and to to go to these all these peoples, we need to go through startups and big pharmas and go uh, fight for this money, because we need a lot of money to start a clinical trial, phase 1, phase 2. And here in Brazil, we don't have this. We don't have this history, right? It's not common. We have just one venture capture specialized in biotechs that we are talking already. It's Vesper, and we are talking with them. Just one. I know that in US, if you go to California, there's like hundreds venture captures there. So, but beside all that, we are going a good way. We are getting everything here, but I know that sometimes we will have to go abroad and continue our work that started here there, because there maybe US there has all the big pharmas and everything that we need to get it to the patients. My main motivation is get out these to the patients because they need it the most.
Absolutely, absolutely, and yeah, I mean, you're definitely you're onto the right path. You clearly have a very impressive team, no doubt. And yeah, you know, whether resources are available in Brazil or not, yeah, I mean, it's just a plane ride away to, I don't know how many biotech VCs we have up here now in the US, a couple thousand, but you'll definitely find the money for this, and no doubt. And it's,
Yeah.
I'm rooting you on. As I said, it's clearly a very elegant approach, and you know what you're doing. But, you know, I guess separate from now the company, and you're going to be road showing on business, any other interesting things coming up on the sort of the university side? Yeah, any conferences you're going to be attending, any lectures or so forth that we should know about that you want us to link to? Any other hot topics that you might want to mention while I have you today?
Well, no, I still a PhD fellow here, so we do a lot of work in the university. I will talk a little bit about our work in a congress, immunotherapy, about Uniparity and COVID in Barcelona next August.
Okay.
Yeah, and I will also participate in Oncolytic viruses Congress, a meeting in Boston, that it will be in October. I'm really looking forward to this Congress because this new oncolytic therapy is not, there's not a lot of research is working, so it's a small meeting, and I'm really looking forward to meet all these people that are being very doing the same as I do, right? Turning the bad guy a good guy. And I think that's all. I'm very happy to make a virus, that a Brazilian virus, right? A Brazilian problem here in Brazil. And I like to think this as a gift. I know that there is a lot of suffering in the beginning, but the main purpose, of we as a scientist is this, is make it resolve the bad problems and maybe turn it a good thing.
Yep.
Right? By studying, by figuring out the mechanisms and everything. So I feel like very, very, very happy. Like it's a gift that comes from this Brazilian, and now I'd like to return this gift for these same kids that need and are dying with cancer.
It's a great message. It's a wonderful story, and really wishing you and the team the best with this. I think it's a positive thing that there are conferences on oncolytic viruses nowadays. There probably weren't
Yeah.
any in the past. So I mean, it shows that the area, like a lot of these new technologies, is coming of age, and that's great for you as well. So for once again, wishing you the best. For everybody that's going to be listening to this podcast across the networks or watching on our YouTube channel. You've been listening to Dr. Caroline Kaid, co-founder and chief science officer of Biotherapeutics, also postdoc fellow, Center for Human Genome and Stem Cell Studies at the University of Sao Paulo. Dr. Kaid, I want to once again thank you for taking the time out of your business and research schedule to come talk to us. Thank you for everything you're doing with this research. And as we say here on our show, thank you for helping to create the better tomorrow through this research. Very, very inspiring and exciting story.
Thank you. Thank you, everything. And thank you for all you that listen. If you got it until here, I'm happy that you listen to the story. Thank you. And if someone has any question, you can contact me in social media and everything.
Yeah, we'll put the links.
I'll be happy to answer any question.
Wonderful. Wonderful. Thank you.